Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. Now the purpose of these videos is to provide a basic understanding of some of the fundamental principles in microbiology. If you're taking an instructor that goes into more detail or into different topics or explains things differently, please make sure that you understand all the material that your instructor wants you to know at the level that you want to know it. I do want to apologize. I know these videos are rather crudely done and they're being done under some duress. I have a short period of time in order to get these videos out for my students while we're in this COVID lockdown. So if they're not necessarily the most technically brilliant videos as far as production is concerned, there's no editing done, please forgive me. I have a cell phone and a tripod, I got a marker board and me, and I'm trying to do these in a short period of time to meet the deadlines for my students to stay on top of their materials. If you do find them helpful, please hit like and or subscribe or leave me a comment. My goal is to reshoot all these videos and make them a little bit nicer and sleeker and shorter and more concise with less ums and ahs. But again, I'm doing these all sort of as an impromptu one-take video. So be a little bit patient or kind with me in that regard. Now, what we've been talking about in the micro course so far is we've been talking about the different types of living organisms. We've talked about microbes. We've talked about ways to observe them like microscopy and staining. We've talked about microbial metabolism and microbial growth. And the reason we need to understand all of that stuff is that we also need to understand how can we control microbial growth. Many microbes cause things like spoilage. They cause pathology, infection, disease, and death. And we need to understand how we can control their growth so that we can decrease spoilage of food or preserve foods. We can prevent the contamination of things like feeds and soils that can be harmful to crops or to cattle and things that we're trying to feed. We can also, the main goal for us, since this is a health science related course, is to prevent and or understand and treat infections, okay, and pathology. So and also design treatments, the proper treatment, if we understand what is causing the pathology. We're going to focus a lot on bacteria. I very often will refer to bacteria. Sometimes I really mean to say microorganisms because eventually in this course we're going to get into things like fungi, parasites, viruses, and the immune system and how our body fights this. But when our body is unable to cope with an infection and the infection is overwhelming our immune system, sometimes we need medical intervention. The use of drugs and chemicals and compounds and other treatment types of treatment in order to prevent, fight, and cure infections. So that's sort of our goal of understanding. Now, in order to understand the control of microbial growth, there are some definitions we have to use. And so I'm going to go through quite a few definitions. Please forgive me if I look over at my notes from time to time. I just want to make sure I'm going over them because I'm doing this off the top of my head. I want to make sure I'm going over them in a specific order for those who are in my class and they have some notes in which to write these definitions. Okay, so one of the things we want to talk about is the definition of sterile. Okay, and sterile simply means the absence of living organisms. Now you could look up sterile, and sterile might mean the inability to reproduce. But you also got to think of these definitions sometimes in the context of microbiology. So this simply means the absence of living organisms, and in particular we're talking about microbes. When something is sterile, that means I've killed all the life forms. And so the sterilent is any agent used to sterilize something. Now I say the word something, I could use some more technical language, but these are somewhat a blend between the technical definition and what I call the Mr. Long definitions so that we can wrap our brain around it. But we use a sterilant to sterilize. That sterilant could be a chemical like alcohol or phenols or an antibiotic, or it might be some physical characteristic like heat or UV radiation. So whatever the sterilant is, the goal is to remove the presence of any life forms so that we can prevent contamination, prevent spoilage, prevent pathogenicity, and other things. Now, if I do sterilize, I mean, these are all related terms. So to sterilize simply means to remove all living organisms. And we're going to look at some things that we can do to sterilize things. And the point of controlling microbial growth is not always sterility. We don't always want to sterilize things. It's not always necessary. For example, dairy products that we consume very often have microbes in them. But we want to get rid of the harmful microbes and those microbes that can cause things to spoil, or we want to control the number so we can prevent spoilage. Some foods have preservatives in them that slow the rate of microbial growth to slow spoilage, but eventually things are going to spoil. And so in a health context, we want to talk about sterilization quite a bit because a lot of the instruments used in surgery need to be sterilized. Sometimes the gown and the clothing that you need to be sterile so that so that you don't prevent or introduce a pathogen or infection into the environment. Now, when we talk about commercial sterilization is not always designed to remove all living organisms. It is designed to sort of reduce the number to decrease spoilage. So commercial sterilization is not something that we're necessarily interested in this course, but we should mention it. When we talk about disinfectant, then that is slightly different definition than a sterilant, okay? So when we talk about a disinfectant or disinfection is the removal of harmful microbes. We're trying to remove whatever can cause the infection. We don't care about those microbes that are harmless. So disinfection and a disinfectant is something that is used to remove harmful microbes, but it's not necessarily a sterilant. So that's a big deal when we're talking about sterilants and disinfectants. For example, a lot of hand sanitizers are not sterilants. They're not going to kill all the microbes on your skin. They're going to remove hopefully most of the harmful microbes or decrease the numbers so that the likelihood that we pass on some harmful microbe is decreased, but they're not necessarily going to kill them. That's important for people who have to do scrubs for surgery. And part of the scrub is not going to be to disinfect, it's going to be degerm. So when we talk about something like to degerm, this is the physical removal of microbes from an area. So for example, if someone's going to scrub for surgery, you use a disinfectant, and that disinfectant can be something like povidone iodine solution or betadine solution, which is povidone iodine, or chlorhexidine. And those chemicals can be disinfectants in the sense that they will kill the harmful microbes and even some of the non-harmful microbes. And there's the degerming. So as you scrub, part of the physical scrub and the rinsing is to physically remove bacteria from the surface and wash them away and decrease their numbers. So to degerm something is simply to talk about the physical removal, where a disinfectant is to remove the harmful microbes. To sterilize means you kill all the life forms present. Okay, we can't do that with our skin. To sanitize is to make something sanitary, or we can say it's to decrease the number of microbes to safe levels. Okay? So to sanitize something doesn't necessarily mean that we're going to disinfect or sterilize it, but we're going to decrease the number of microbes to, safe levels or to some health standard. Essentially what this does is it decreases the chance of infection or introducing a spoiling bacterium or something. But one of the ones I want to talk about, instead of just disinfection or degerming or sterility and disinfection, is we want to talk about the term sepsis and antisepsis and antiseptics. So, when we talk about sepsis Sepsis can be defined a number of ways, but when we're talking about microbiology, sepsis usually means the presence of pathogens. So if someone has gone septic, if a wound is septic, that means there's pathogens present causing an infection. And so antisepsis is the absence or removal of pathogens. Now, I know a lot of these definitions are starting to sound very similar, but there are some technical differences, and you should know them. So, antisepsis is where we disinfect usually living tissue. So when we talk about sepsis, this is not only the presence of pathogens, but we're usually talking about in living tissue. And this is the removal or absence of pathogens from living tissue. So if I say that I'm going to have an antiseptic, that means that antiseptic is going to be able to kill any pathogen or bacteria or microbes that are present on the living tissue. Like an antiseptic mouthwash will kill or remove a lot of the bacteria from, or the pathogens that are present in your mouth. Or if we use an antiseptic on a wound, and sepsis simply means that I have pathogens present in the wound. So if a wound has gone septic, you know there's pathogens present and it's causing some toxins to build up. Now, when I talk about the term side, anytime you see side at the end of something, it simply means death or to kill. So if I talk about a bactericide is something that's going to kill bacteria. Now, I'm not going to write all of these down, but if I talk about a fungicide, that's something that kills funguses. If I talk about a viricide from virus, it's something that can kill viruses. So anytime we see side on here, we're talking with CIDE, we're talking about something that causes death or kills something else. A germicide is something that kills germs. Okay? And we're going to be looking at different types of these in just a minute. Let me make sure I've covered all the definitions I wanted to cover and I haven't forgotten any. I don't have my notes in front of me. Oh, one last term I left off. When we talk about something as being static, or we talk about the term stasis, that means to maintain a low number. Okay? Or basically it's something that stops microbe growth. It doesn't necessarily kill them, but it keeps the number the same. It holds them at bay. So when we talk about something being bacteriostatic, what that means is for some reason, it's not going to kill all the bacteria. It's not a bactericide, but it will prevent the bacteria from growing in number and hold them in a holding pattern. Okay? So those are some terms that I want you to know. Now, when we talk about controlling bacterial growth or microbial growth, a lot of times we're going to talk about killing bacteria or keeping their growth at a certain number, things that are bactericidal or bacteriostatic. And there's a number of things that we can take into account. Some of the environmental factors that can affect this. So we can talk about environmental factors affecting the control of their growth. One of them is the time of exposure. How long does it take for this particular substance to wipe out all the bacteria? How long do I need to expose a microbe to some chemical to wipe them out? So we talk about, the sort of death times and other things, and we'll talk about some of these in just a minute. Another environmental factor, by the way, is something like the number of microbes present. This can also affect the exposure time. If I only have a few, then a little bit of the chemical could wipe them out. If I have a whole bunch, then the chemical needs to sit long enough so that all the bacteria get exposed to it and they can be wiped out. Okay. And another thing can be the characteristics of the microbe involved. You know, some microbes are resistant to heat, or some microbes can withstand a high salt concentration. So salt as a preservative may not work on all microbes, or the particular type of heat and how long we expose them to the heat. So it's the time of exposure can also be determined by the number of microbes present, some of the physical characteristics of the microbe, their hardiness and exposure to acid or base or heat or cold or whatever it is that we're trying to use to control them. So we need to keep this in mind sometimes when we're treating things for stasis or for killing microbes. Now, we're going to look at some different ways that we can do all of this stuff, and one of the most common ways of killing microbes in particular is heat. So we're going to look at, well, before I get to this, let me talk about some different ways that the modes of treatment, whether it's chemical, whether it's physical or whatnot, can kill bacteria. So how do these agents affect bacteria or microbes? Well, one is to disrupt the cell membrane. So bacteria have a plasma membrane, and it's made up of lipids with proteins, and those proteins are capable of transporting substances across the cell membrane like ion channels and whatnot. And then the bacteria have an outer covering called the cell wall. Okay? And then even some bacteria can have an outer membrane outside of that. And we've talked about the proteoglycans that make up the glycocalyx and all of this stuff. So if I have something that disrupts the cell membrane or the cell wall, and those are two different things, then I can affect bacteria. For example, most cells are not tolerant of high salt, but some bacteria are. And so if I have a hypertonic solution or a hypertonic environment, and I put a lot of salt outside the cell, one of the things that can happen is we can draw all the water out of the cell by doing this. And what can result is I can get a bacterium where the inner cell membrane starts to separate from the cell wall and they shrivel up. We call this process plasmolysis. And plasmolysis can slow the rate of bacterial growth. It disrupts a lot of their metabolic function. But one thing that can happen sometimes is we can have things that damage the cell wall. And so if I disrupt either the cell wall and or the cell membrane, then I can affect bacteria. It turns out that bacteria sometimes are hypertonic to a lot of solutions. And so if I have a hypotonic solution or a hypotonic environment, then water wants to flow in where the higher salt concentration is. That would cause the cell to swell and rupture. But because bacteria have a very rigid cell wall, a lot of bacteria are not susceptible to what we call lysis or cytolysis, the rupturing of the cell. But if I have chemicals that can disrupt the cell wall or the cell membrane, then I can kill bacteria. And so, for example, one of the enzymes that's present in a lot of our secretions and saliva and mucus and tears and some of our sweat is called lysozyme. And lysozyme can disrupt the cell wall of bacteria. They can take on a lot of water, and therefore the cell can pop or rupture, and we call that cytolysis. Another way that chemicals can disrupt the cell membrane is to bind to proteins present in the cell membrane and prevent the transport of certain substances into or out of the cell. So many of the agents that we're going to use to affect bacterial growth can disrupt cell membrane or cell wall function. Okay? And we can either slow their rate of growth or kill them depending on what we do. And you need to know the terms plasmolysis and cytolysis and why they occur. Now, another way that we can affect bacterial growth is we can disrupt nucleic acid function. When I say nucleic acid, I'm talking about DNA or RNA. We can mess up their structure or function. So we know that heat can melt or denature proteins. Heat can also denature DNA, so to speak, in that it can break some of the bonds and mess up the physical structure of both DNA and RNA. We can literally sort of melt the DNA or melt the RNA. That would disrupt cell reproduction, or it could disrupt protein synthesis, disrupting metabolic pathways, which would slow, prevent, or bacterial growth or kill the bacteria. So we can disrupt nucleic acid structure. We can also disrupt protein function. So we know that we can denature proteins. So heat can denature proteins. Heat can also cause some proteins to sort of stick together or clump together after a while, that we call coagulation. If I disrupt the function of a particular enzyme, and some drugs can do that. Some antibiotics can simply disrupt a particular protein function, either in the outer membrane of a bacterium and prevent it from transporting certain substances needed for survival, or they can literally go in and disrupt specific enzymes. So if I have a drug that can affect the enzymes that are functioning in DNA replication and therefore microbial reproduction or viral reproduction, then I can stop the growth, slow the growth, or particularly kill them. So we can affect microbes in a number of ways. Disrupt cell wall or cell membrane. In viruses, we can disrupt the protein coat. We'll talk about that when we get to viruses. We can disrupt nucleic acid structure. We can disrupt protein function, and that we denature enzymes or disrupt the transport of certain nutrients into and out of the cell, and the use of those nutrients. So, now, we're going to talk about the physical methods that we can use to do some of this. And I think I'm going to stop after the physical methods. I might, and we'll do chemical methods in another video. Try to keep these short. I might get on a roll and keep going. Who knows? So some of the physical methods, these are physical methods to control microbial growth. One of the most common and easy to use is heat. As we know, heat can melt DNA or change the structure of DNA. It can denature proteins and really disrupt bacterial growth. And even sometimes under direct heat, we can simply incinerate, burn the bacteria to nothing but some carbon. So we'll talk about some of these methods. For example, when we use direct heat, that would be like flaming your loop, or an incinerator at a hospital where they take all the biological waste and they simply just burn it down to ash. That's obviously going to kill all the microbes. So heat is an excellent way. So when we talk about heat, one way to use heat is direct heat. And we call that incineration. You simply burn things and reduce them to ash. That'll remove life forms for sure. Now, sometimes when we talk about the use of heat, we can talk about boiling. This is sort of what we call a use of moist heat, but it's not necessarily steam. So in boiling, we simply heat in fluid, right? And if I heat the fluid high enough to boil, well, those temperatures are going to be high enough to do a lot of denaturation. We can denature proteins and enzymes, disrupt DNA function. And so boiling is really, really good. Although some organisms can withstand high temperatures, many of them cannot withstand direct heat or incineration. Okay? Another way is what we call autoclaving. An autoclave is simply a special oven that uses heat, pressure, and steam over time. And one of the nice things about autoclaving is as I increase the pressure, I lower the boiling point. I can increase the effectiveness of the heat, so to speak. So I don't necessarily have to heat things so high in order to kill them. And the steam can penetrate nooks and crannies. And as you know, moisture absorbs heat and carries heat very well. So the steam entering all these little nooks and crannies can also carry the heat in. And the pressure helps force the steam and the heat into the substance. So I can use an autoclave under pressure with steam at higher temperatures. Now, one of the advantages to boiling is that it can kill a lot of organisms. One of the disadvantages of boiling is that sometimes I don't want to get things wet. So sometimes we simply just have to heat them in a dry oven. That's not always the best method. One of the advantages of autoclaving is I don't have to necessarily always get to the same height of temperature in order to kill the microbes, and can be used for a lot of things like glassware and clothing and things to sterilize it. Now, we can talk about things like the thermal death point and the thermal death time. And so when we talk about heat, we have to discuss things like what's called the TDP, or the thermal death point. Okay? When we talk about the thermal death point, one of the things that we're looking at is the lowest temperature at which all the microbes are killed over a period of time. It's usually about 10 minutes. So this is the minimal temperature at which, now these are abbreviations, the minimal temperature at which all the microbes are killed in about 10 minutes. And this can be, you can study this, and you can do tests of, say, E. coli and different microorganisms. Say, what is the thermal death point for this microorganism? How long does it take to kill all the E. coli present in something in 10 minutes? Or at what temperature do I minimally have to heat something to kill all the E. coli in 10 minutes? Or all of another particular bacterium like Bacillus subtilis or Klebsiella pneumoniae. So every bacterium has a certain thermal death point, and if you know that, then you know what temperature you need to use over a period of time to sterilize this particular substance. We can also talk about the TDT, which is the thermal death time. Okay? So when we talk about the thermal death time, one of the things that we need to talk about is how long does it take to kill all the microbes? And essentially what we're looking at here is the minimal amount of time it takes to kill all the microbes in a liquid at a given temperature. So one of them is looking at as the minimum temperature, the thermal death point, the point at which death is caused at this temperature, okay? Or at which temperature do we kill the microbes? The thermal death time is, if I know a specific temperature that I can treat something, how long does it take? Here we know the time is determined. Now we need to figure out what the temperature. But for thermal death time, we need to know how long it takes to kill something at a known temperature. Okay? And finally, one of the things we can look at is the decimal reduction time, or the D value. Okay? So let me erase all this to make some room for myself. And we're going to talk about the decimal reduction time. Which is also called the d-value. Which is not the same as to devalue something. To devalue it means you decrease its value. This is known as the d-value, as an abbreviation, okay? So, when we define the decimal reduction time, what we're talking about is the number of minutes it takes to kill 90% of the microbes at a given temperature. So, this is the amount of time to kill 90% of the microbes at a given temperature. Okay? So there's a number of ways we can look at how we treat something when it comes to using heat. The thermal death time, the decimal reduction time, and we can look at the thermal death point. Now, another physical characteristic that we can use to control microbial growth is going to be moist heat. We've talked about boiling, we've talked about autoclaving. Boiling does not kill all microbes. Autoclaving can reduce the amount of the amount of time it takes to kill bacteria due to increased pressure. And we can talk about pasteurization. Of course, this is named for Louis Pasteur, and basically Pasteur used heat over the gooseneck of a particular type of flask to prevent microbes from coming into a sterile environment, and showed that he basically put an end to this idea of spontaneous generation. Well, people took that, including Louis Pasteur himself, and basically used the idea of temperature to kill microbes. Now, when we talk about pasteurization, it does not necessarily sterilize something, okay? It's not using the same level of heat. There are some products that if we heated them to a boiling point or autoclaved them, we would cause damage to the product itself, like milk. We can't boil milk or it will start to curdle, and then it would not be any good. So pasteurization doesn't necessarily sterilize, it decreases the number of microbes to be, to safe and healthy levels. Now, what we can do when we do this is, if we decrease the number of microbes, we slow the rate at which spoilage would occur. And this can be done for dairy products, it can be done things for like beer and alcohol products. So you hear about pasteurization all the time, and now there's even a process called cold pasteurization, and this doesn't necessarily kill the microbes. What it does is it slows their growth rate so that it decreases their number over a period of time. Now, when we talk about using heat, sometimes it's sort of the absence of heat. So another thing that we can do is we can use refrigeration. Again, refrigeration does not necessarily kill microbes. What it does is it can decrease the rate of microbial metabolism. It slows the rate at which microbes can make energy, therefore slowing down their reproductive rates so that it can delay spoilage and prevent microbial growth over a period of time. Now, there are some organisms, there's one organism that is somewhat resistant to refrigeration, and it's called listeria. So if you've heard of listeria poisoning, boy, that's some bad stuff. It leads to a really bad food poisoning. And finally, there's a thing called freezing. And basically, freezing can decrease the rate of microbial metabolism as well to really slow levels. Now, there are some bacteria that can enter what we call a spore form, and so it doesn't always kill those bacteria that sporulate, but it kills most bacteria. It can kill many bacteria. Okay, this is my abbreviation for bacteria. I don't always like writing the word out, especially for this class. But it can kill many microbes. Essentially, when water starts to form ice crystals, the ice will expand. I don't know if you've ever seen this, but if you take a bottled water that's pretty full and you put it in the freezer, the bottle puffs out or even cracks and ruptures because the ice, as water freezes, the molecules expand. And so if they do expand, they can sometimes rupture the bacterium or the microbe and kill it. So all of these are ways using a temperature. I say heat, but we can use temperature to control microbial growth. One other physical characteristic that we can talk about is radiation. Okay, so these are all using sort of heat or temperature to control bacterial growth, killing microbes or slowing their rate. You also need to know about radiation. So radiation is basically using high energy particles. When we talk about light, visible light and even non-visible light fires energy out in little bundles called photons. But there's other types of particles that can be released, like X-rays and gamma rays. So we can talk about different types of radiation. One type is UV light. So essentially, we know that photons travel at a certain wavelength. And specific photons can have higher or lower wavelengths. The shorter the wavelength, so if I measure the wavelength between any two waves of a particular photon, then all of the lengths of every wave would be the exact same distance for one photon. A separate photon might have a smaller wavelength, and so I might fit 3 cycles in where another one only gets 2. So we can talk about the frequency or the wavelength. Now, one of the things that we know is that the lower the wavelength means the higher the energy at which the photons can travel. And the more energy at which they travel, when they hit other molecules and atoms, they can disrupt them. They can dislodge electrons. We can shatter atoms like in atom smashers when we do studies in physics. And so high energy radiation or high energy light can damage things. Like we know that ultraviolet light can damage DNA. It can cause these things called thymine dimers. And that can disrupt enzyme production or protein production. It can also disrupt cellular reproduction, binary fission, the replication of DNA. And so ultraviolet light can damage DNA, and that can disrupt bacterial growth. Doesn't always kill bacteria, but it can if the radiation is high enough to damage sort of enzyme function or enzyme production. So, as you know, ultraviolet light is traveling at a much higher wavelength than is visible light, and therefore it's very damaging. One of the problems is it's also damaging to human cells. It can cause skin cancer. It can disrupt our DNA in our skin cells and result in skin cancer. So exposure to too much ultraviolet light, no matter what tanning bed manufacturers tell you, ultraviolet light damages skin, and it can essentially damage cells at the DNA level and result in cancer. I'm sure one of them wants to get a lawyer available to repudiate that with me. Now, one of the problems with ultraviolet light, though, is often it only kills bacteria that are growing on the surface of things. So it can only, it cannot kill bacteria that are, say, growing in our gut. Now, there's a few other types of radiation that we can talk about, and one of them, in addition to ultraviolet light, is called ionizing radiation. And ionizing radiation can include things like X-rays, which are very high energy, and we can talk about gamma rays. These are super high energy, and these can do damage to a living tissue, and they can really do damage to human cells, so they're not very good. But one of the things that they're good at is treating products that are packaged. So we can use ultraviolet light or ionizing radiation on things that are packaged, like when you're opening a sterile gown, a lot of times they've been treated with some kind of ionizing radiation to kill any microbes inside the package. So as they're being manufactured and sealed, all these sterile products that you will open into the surgical field, or when you're opening a sterile package that has a syringe in it or a needle, very often they've been treated with ionizing radiation. So one of the problems with these is they're harmful to humans, but they are good on non-living, or I won't say non-living products, but inanimate products like clothing and materials used in surgery and in the medical field, tubings and other things. Anything that has to be sterilized, we can often use these things to sterilize them so they don't introduce infection. Now, also, one of the problems with these two is the machines that can develop X-rays and gamma rays are extremely expensive. When I worked for a group of orthopedic surgeons, one of our X-ray machines was several $100,000. And so, it's expensive, but if you have a big enough industry, then you can use them. Another way that we can disrupt bacterial growth is by filtration. We can filter out microbes. So filtration is not always a good way to kill microbes, but filtration can decrease the number of microbes present. You literally filter them out. One good way is through what we call HEPA filters, high energy filters for air conditioners and things. And HEPA filters can filter out a lot of the bacteria blowing around that can make you sick, as well as other debris and contaminants. And there's specific filters that are designed for specific uses, and the smaller the pore size, so one filter might have pores this big, the next filter might have even smaller pores. And these pores are on the level of microns, right? And we talked about microns for those of you that have lab. A micron is a micrometer or a millionth of a meter. And we even have some filters that are on the level of nanometers, really tiny filters. And essentially, if I want to remove a number of bacteria, say from a food product, a liquid, I can run them through a series of filters and filter out all the microbes. We do this in like cold filtration of beers now and other things. And this can be done for a lot of solutions that are used in a setting. Now, one of the problems with this is very often the filters are not small enough to filter out viruses. And so if there are viruses present, they're not really good at getting rid of the viruses. Okay? Another way that we can control bacterial growth is by drying. You hear about things being freeze-dried. Well, in drying, it's simply the removal of water. And so we can dehydrate things like dehydrated meals, MREs, meals ready to eat. For people who are like, preppers for disasters, they buy the MREs. I'm not going to lie, I have a few because I live in a very high hurricane-prone area. But these have been desiccated. So you use a thing called a desiccant. A desiccant is a substance that binds up water vapor. So you see those little silica gel packs, those are desiccants, and they bind up moisture so that moisture can't damage things. And if I remove all the moisture from an area that has microbes, as you know, bacteria really love water, and they need usually a very high water-rich environment in order to grow and reproduce. And so desiccants and drying things can often kill or decrease the number of microbes. So we can use evaporation techniques, remove all the water through evaporation. We can use a desiccant or desiccation. These are ways of drying things out that kill or prevent microbe growth. Another way is through what we call lyophilization. And I mentioned this. To lyophilize means to freeze dry. Now, in this process, essentially what we do is we freeze things. And as you know, when we look at water, water can be in a solid, it can go to a liquid state. If I have some water in a glass, I can freeze it, I can make it a liquid, or if I heat it, I can start to boil it off. So water can exist as a solid like ice, a liquid like water, or a gas like vapor. Well, if I skip this, if I can make something freeze and then make it skip the liquid phase, and very often this is also called sublimation. And in sublimation or lyophilization, basically what we do is we freeze something and then we use a special chamber that will use pressure and other conditions so that when we do remove the water, we can go straight from the solid to the gas phase and skip the liquid phase. And as we remove that water, it will very often kill a lot of microbes. We call that lyophilization, okay? One of the problems, by the way, with lyophilization is the machinery is rather expensive, okay? Now, a little while ago, I talked about disrupting the microbe membrane. And so that's one way that we can also use to slow or kill microbes, slow their growth or kill them, is by using osmotic pressures and tonicity. Many microbes cannot stand a high salt concentration, so a few can, but if I want to kill microbes and I know that the ones that I'm interested in removing are not halophiles or cannot tolerate high salt, then I can use salt and tonicity to kill them, or what we call the osmotic strength. Not only do we use salt, but in this situation we can use a very high concentration of salt or sugars. And very often we do this to preserve things. So a lot of preservatives we use high salt and high sugar concentration. And basically they work by plasmolysis. They pull all the water out of the bacteria, cause them to plasmolyze, and slow their growth or kill them. So we can use osmotic pressure or osmotic strength, essentially using salt or sugar to pull the water out of something. It's different than dehydration by using temperature or lyophilization. It's simply pulling the water out using salt and sugar. And we very, very often use this as preservatives, like salting meat and salting fish for preservation. Now, a few other things we can talk about. And I said I was going to stop the video after I talked about physical methods, but I'm going to finish this up. Chemical methods. How can we use chemicals to control microbial growth? Okay. Well, a lot of things, a lot of different chemicals can be used to either decrease the number of bacteria, to disinfect, to sterilize, to sanitize. And so we're going to use those terms with some of these chemicals that we're going to talk about. And when we talk about this, we also have to talk about time of exposure. We have to look at the bacterial characteristics and essentially how does this substance work? Therefore, how long do I need to use it, or under what conditions must it be used in order to be effective? Now, one of the things that we see, everywhere we go is hand sanitizer. Now that we're under this COVID situation, and people are sanitizing their hands like crazy. And very often you'll read things that say this decreases 99.9% of bacteria and viruses. But if you read the fine print, usually that's on what we call a smooth, non-porous surface. Our skin is not smooth and non-porous, so it's not going to kill 99% of the germs. It will kill some, and it will decrease their numbers, and therefore help prevent the spread, but it's not going to stop it. It's not a foolproof method. Okay, so we'll talk about some of this. Now, there's a group of chemicals called phenols and bisphenols. And very often these can prevent microbial growth. Joseph Lister, one of the guys who started the antiseptic and aseptic technique in medicine, used a stuff called phenol, or some people he called it carbolic acid. Okay? And essentially these are very good disinfectants. Now, we have to look at how effective these things are, and there's a phenol coefficient, and we can look at the time and the concentrations at which it works. But nonetheless, these things are very effective. We also can look at what's called the dilution factors, and we're not going to get into all those details, but I do want to tell you one of the things that we know about this is there's another one that you often see. I'm going to put in parentheses. One of the types of phenols is a stuff called cresol. And this is stuff that's used in treating lumber so that it doesn't get broken down. So when you look at chemically treated lumber, it's very often treated with a phenol or bisphenol called cresol, and it preserves the wood. Now, let's see. I want to make sure I cover everything before I move on. Well, when it comes to the phenols and bisphenols, we can also use the phenol coefficient to test the effectiveness of germicides. And so we could talk about things like the phenol coefficient, where basically we compare the several dilution factors or concentrations of the phenol and how effective it is at killing germs. And we have to check these germicides not only in their concentrations, but over a period of time. And sometimes we use a standard like Salmonella bacteria, and we look at how long it takes to kill the Salmonella typhi. Sometimes Staphylococcus aureus is used. We can talk about the concentration or dilution factor of a germicide, and this is basically how effective it is compared to the phenol coefficient. And there's numbers. If it's less than 1, it can tell you that it is less effective than phenol. So when we talk about phenols, we talk about the phenol coefficient. If something has a phenol coefficient less than 1, that means it's less effective than the phenol. If it's greater than 1, then it is more effective than the phenol. Okay? So sometimes when you look at the effectiveness of a germicide, you can look at its phenol coefficient and tell me if it's really, really good at killing the germs or if it's not. Again, this is all relative to a certain time called the phenol coefficient, the time and concentration. I don't want to get into those details. Another way that we can look at how chemicals are effective is we look at what's called the disc test or the paper disc test. Okay? And some people call this the Kirby-Bauer measurement. Okay, the Kirby-Bauer test. And essentially what happens here is you have a plate of bacteria and they're growing. And you take these little paper discs that are soaked in whatever agent you're interested in studying, a particular antibiotic or antiviral. And if you put these little discs on here where the growth is, if you start to see a big ring where there's a lack of growth, and let's say I have a disc that has one substance on it, and I put that on a plate, then I can see at the kill zone the area where there's no growth, and another disc, I may not have any effect, and you wouldn't usually necessarily do these on the exact same plate, but I don't want to make multiple drawings, and another one might have a smaller area. So we can look at the rate of or the lack of growth and the size of the ring based upon whatever compound or substance this disc is impregnated with, and they call this the Kirby-Bauer test or the paper disc test. It's very often done with antibiotics, and you take one of these soaked in antibiotic and put it on a plate and see if it kills the bacteria. And sometimes they're very effective at killing the bacteria. Sometimes they only kill some. They're not as effective, and we could look at which antibiotic is effective against which bacteria, and therefore we can prescribe specific antibiotics for specific treatments. So that's the Kirby-Bauer or disc test. And you look at these clear zones in order to, and the clear zone or the death zone is very often referred to as the zone of inhibition. That's the area where it is inhibited growth of the bacteria. And then we can also look at what are called use dilution tests. And basically these use several levels of dilution of the agent. And so when we use the phenol coefficient, we can look at how effective something is compared to phenol at a certain concentration and over time. We can look at the Kirby-Bauer test or the paper disc test. How effective is it at killing microbes on a plate? And we can also look at the use dilution test. And essentially what this does is we take a certain amount of a substance and we know what the concentration is or the dilution factor. So I can have like a 1 to 5 dilution, I can have a 1 to 10 dilution, a 1 to 20 dilution. So the amount of substance that is in water, and I can look at say 1 to 1,000. We can use several levels of dilution, and essentially what I do is I inoculate every one of these, and this is one substance, maybe one antibiotic or one chemical, one germicide, and I say at a 1 to 5 dilution, is it effective? At a 1 to 10, at a 1 to 20, a 1 to 100, 1 to 1,000. And I look at which dilution rate at which it best kills the microbes. And then that's what I want to use in order to, when I bottle or sell this product, or use it as an antiseptic or germicidal agent. And so all these different tests are sort of how we determine the effectiveness of the chemical methods that we're talking about. Now, and one of the things that I guess I can tell you is when we talk about the use dilution test, what we want to find is what is the highest concentration or the lowest dilution factor at which the fluid remains clear or not turbid when we inoculate it, and you can either look at the agent and a specific microbe, or you can look at different microbes with the same agent at different dilutions, and I can say at this dilution it kills these bacteria or germs, at this dilution it's more effective at killing these, and then we can target that product to certain things. What you need to know about the use dilution test is basically it looks at the effectiveness of a germicide or antimicrobial agent at different dilutions, and we find the dilution at which it's most effective. Okay? Now, again, all this stuff will be typed up. If you're in my class, it'll be typed up in some notes, or it's covered in your textbooks. Now, I do want to talk about one or two other things, but I do know that we're kind of getting sort of to the end of an hour or so, maybe even longer. So I'm going to go in and talk about the specific types of chemicals that we could use in order to kill microorganisms. And again, some of these agents are going to do things like damage cell walls and cell membranes. Some of them are going to disrupt DNA function. There's intercalating agents that fit in between the bases and disrupt DNA replication and protein synthesis. We have chemicals that can bind and inhibit enzymes. We've talked about enzyme inhibition when we talked about microbial metabolism, competitive and non-competitive inhibition. We have substances that can disrupt the transport of certain molecules across the cell membrane or bind up certain molecules. There's a number of ways that these chemicals work. I'm just going to mention a few, and I'm going to mention them briefly. I'm actually going to put my notes in my hand just so that I can make sure that I run through them quickly and not spend too much time. Okay, so one of the things that we know about phenols and bisphenols, not only are they used to sort of test the effectiveness of other agents, and there's another one called biguanides. The biguanides are not used in order to test the effectiveness of other agents. But one of the things that we know about them is that these can disrupt membranes. They disrupt the plasma membranes of many microbes, okay? They can dissolve lipids, and as they dissolve the lipid membrane, it will allow the cell to essentially be broken up, and it allows substances well, it's basically like cutting someone open. Everything spills out. It would also allow other harmful substances in. We can use things called halogens or halogens. If you've taken chemistry, you know what halogens are, the halides. And we're talking about things like iodide. We're talking about iron, copper. We're talking about some of the halide compounds that can, essentially these things can disrupt enzyme function. And really iron's not a very good one, but iodide, copper, zinc, and a few others, mercury can disrupt enzyme function. And actually, some of them fall under what we call the heavy metals. They're not exactly halides, but they're known as the heavy metals. And I'm not talking about metallic. I'm talking about things like mercury, which is abbreviated as Hg, things like zinc, and a few other heavy metals that are effective at damaging proteins. So these can also disrupt protein function, either enzymes or things like membrane proteins, and bind them up and prevent the transport of stuff. So Phenols and phenols, bisphenols, and biguanides can disrupt lipid membranes and dissolve the lipid membrane. Halogens like iodine and copper can disrupt enzyme function. Heavy metals like mercury and zinc can disrupt protein and enzyme function. Okay. Alcohols. So when I talk about alcohols, we're talking about things like isopropyl alcohol and rubbing alcohol, which you can put on a wound. It's not very good at killing a lot of things, but one of the things that we know that alcohols can do is that they can denature proteins. And very often, certain concentrations of alcohols are really effective. That's why we use certain concentrations when we treat things. One of the things we know about alcohols is it's not really good for wounds, though, because it can also damage some of the healthy cells. It can dehydrate cells as well. There are things called surfactants. Surfactants very often can damage cell membranes. And certain surfactants are good at killing microbes. We do have a surfactant in our lungs that prevents our lungs collapsing. And one group of chemicals are called the food preservatives. And I'm not going to go into any of the specific chemicals. There's a long list of food preservatives. But essentially, a lot of the food preservatives can decrease microbial growth. They're not always good at killing the microbes, but they can just slow their rate of growth so that they can prevent spoilage. We certainly know that antibiotics, very often these kill bacteria. Antibiotics can work in a number of ways. We're going to get into how antibiotics work. And anytime I have the word anti and I have something on it, like an antifungal is going to kill fungi, and antiviral will kill viruses. And very often when someone has a viral infection, it's not necessarily good to give someone antibiotics because antibiotics kill bacterial infections. So you can have pneumonia that is caused by bacteria, and you can have pneumonia that's caused by a virus. And very often viral infections are hard to treat after about 48 hours. So if you're going to take antivirals, very often you need to take the antivirals within a small window of time in order to increase their effectiveness. Things like Tamiflu and other antiviral drugs. There is a certain, what we call the side groups on certain chemicals. So when we look at a chemical X, if it has an OH, we call that an alcohol. If we have an OH, COOH, then call that a carboxyl group. Well, there's certain side groups called aldehydes and ethers. And one of the things we know about aldehydes is that they're very bactericidal. Okay, so when we can very often use these compounds called aldehydes. Formaldehyde is one that's very good at preventing things from rotting. We use formaldehyde to preserve specimens and anatomy. Aldehydes are very much bactericidal. They kill a lot of bacteria. They cannot grow or survive in high aldehyde concentration. Okay. We also know that a lot of aldehydes are virucidal. They kill viruses. The problem is they can also kill a lot of healthy cells and pickle you, so you don't want to use aldehydes on a human being except for in very minimal and specific conditions. All right. And finally, the last one I want to mention is oxygen and peroxygens. For example, peroxide is a peroxygen. So we talked about water, dihydrogen oxide. But when I have one hydrogen for each oxygen, H2O is called hydrogen peroxide. I've got one hydrogen for each oxygen. And essentially, this will release the free oxygen or the extra oxygen molecules. And when I combine two of them that have been released, I get water and I get oxygen. Okay? So if I have two of these, I can get two waters and two free oxygens. And anaerobic bacteria don't like oxygen. And so peroxides in high oxygen environments can prevent the growth or even kill anaerobic bacteria, and it can slow the growth of certain anaerobes. So oxygen and peroxides are very good at killing anaerobes. And very often, some of our pathology is created by anaerobic bacteria. Now listen, this is sort of a broad, quick overview of a lot of information. If I tried to go into detail into every one of these, it could be overwhelming. I could lecture for an entire week on every one of these things that we've talked about as far as the physical and the chemical means. I could go into great detail into how ultraviolet light and gamma radiation and x-rays can kill microbes and damage cells. But then the course becomes overwhelming. We already have microbial metabolism, which in itself is overwhelming. And then microbial growth has got a lot of stuff in it in the chapter. The nutrients and all the requirements to grow bacteria, which also leads to ways that we can control their growth by limiting nutrients, and also all these other ways that we've talked about controlling microbial growth. So we have a lot of information to digest and consume, so I'm not going to go into any more detail. I hope that was good enough for you. I hope at least it lent some understanding to what we are talking about, what you're covering in your textbook. And the list is a little longer than this, but some of the ones that we could talk about are used in industrial uses for feeds and grains and soils. And that's not our interest. Our interest is in the allied health sciences. Although those things can impact our health if we get exposed to them sometimes, but I'm not interested in discussing them in great detail. So I hope you learned something. I hope this covered the basics for you. I hope you had as much fun as I did. Hope to see you in the next video. All right? Thanks for watching.